Okay, so welcome back to this next video in which we are discussing phospholipase C beta enzymes. Okay, right, so at the moment we are in the process of discussing the reaction that phospholipase C enzymes, all phospholipase C enzymes, are going to catalyze, not just the phospholipase C beta enzymes. Okay, right, so we want to see the structure of phosphatidylinositol 4,5-bisphosphate, which is a member of a wider collection of molecules known as phosphoglycerolipids. Okay, now phosphoglycerolipids are all derived from the molecule phosphatidic acid, which we've discussed in the previous video, which consists of a glycerol molecule as its backbone with two long-chain carboxylic acids esterified to the alcohol groups that come off the first and the second carbon. Okay, and then a phosphate group attached to the alcohol group of the third carbon via a phosphoester link. So, how are we now going to modify these phosphatidic acid molecules to create a more general phosphoglycerolipid? Well, the answer is we're going to stick an additional group of the phosphate group here within the phosphatidic acid molecule. Okay, so in blue here, we're going to add in an additional group. Now, how are we going to do this? Well, you remember me telling you that in phosphatidic acid, we still have this existing group here where we have a phosphorus atom double bound to an oxygen with an alcohol group. Okay, so phosphate groups have two of these effectively, and this group circled in blue is now capable of reacting with another alcohol group basically on another molecule. Okay, so you can bring along some molecule that has an alcohol group, and what you can do is you can remove the alcohol group again from the phosphate group, remove the hydrogen from the alcohol group, okay, and then bind this phosphorus atom to the oxygen atom of the alcohol group, again in a phosphoester link. So you can link this phosphate group of the phosphatidic acid molecule to another alcohol molecule, basically. Well, a molecule with an alcohol group in it, okay? And this is how you create a more general phosphoglycerolipid. So the family of phosphoglycerolipid molecules are molecules where you take a phosphatidic acid molecule and you attach on different alcohol group containing molecules onto the other side of this phosphate group here. Okay, right, so the one we are going to be interested in is attaching on a molecule known as inositol. Okay, so we want to see the example of a phosphoglycerolipid called phosphatidylinositol, and well, phosphatidylinositol 4,5-bisphosphate actually. But I want to stress that the phosphoglycerolipid is a much larger family. There are many other things that you can attach on there other than inositol. But we're specifically interested in phosphatidylinositol 4,5-bisphosphate. So we're going to attach on inositol that has another two phosphate groups attached to it. Okay, so we want to see the structure of phosphatidylinositol 4,5-bisphosphate. And a running theme in the subject of phosphoglycerolipids is that the naming of phosphoglycerolipids always begins with this prefix phosphatidyl, and what that effectively means is a phosphatidic acid molecule is going to be within this structure. Okay, and the next portion tells you what you've added onto the other side of the phosphate group of the phosphatidic acid molecule. So in this case, we've added inositol 4,5-bisphosphate. Okay, so all of the phosphoglycerolipids have this first prefix here, which is phosphatidyl. Now, a few little bits of nomenclature then. Phosphatidylinositol 4,5-bisphosphate is often just abbreviated to PIP2. Okay, and this stands for phosphatidyl, that's the first P here. Okay, then the I is for inositol, and then the P2 stands for phosphate, and then the 2 is for bisphosphate, so a phosphate group coming off the fourth and the fifth carbons. Okay, now to be a little bit more specific, some people will write phosphatidylinositol and then they'll put brackets 4,5P2, okay, to avoid confusion, because you can have the phosphate groups coming off different carbons, and this name wouldn't tell you that specifically they're coming off the fourth and the fifth carbon of the inositol ring. Okay, some people even go further, they abbreviate this instead to PTD for phosphatidyl, okay, and then they abbreviate inositol to INS, and then they'll have 4,5-P2. So those are three names that you can see phosphatidyl inositol 4,5-bisphosphate abbreviated to. Okay, so let's now discuss the structure then of phosphatidyl inositol 4,5-bisphosphate. So firstly, let's start off by just sticking an inositol molecule onto a phosphatidic acid molecule to create phosphatidyl inositol, and then we'll modify it by sticking phosphate groups off the 4th and the 5th carbon to create phosphatidyl inositol 4,5-bisphosphate. Okay, right, so let's start with inositol then. Okay, so basically inositol is again an old name for a molecule that would now be called cyclohexane. Okay, 1, 2, 3, 4, 5, 6 hexol. Okay, so this is the correct name, well, the new name for for inositol. Okay, and yes, cyclohexane 1, 2, 3, 4, 5, 6 hexol is a mouthful. Okay, but it's a useful name because now we know exactly what we're working with here. Okay, so we know, and I think I'll go over onto the other page. Okay, we know we're dealing with a 6-membered carbon ring. Okay, that's the cyclohexane portion of the name. Okay, hexane means 6 carbon structure. Okay, cyclo means you need them in a ring, and we're going to have single bonds between all of these carbons. Now, I'm drawing a skeletal formula here, so you don't show carbon atoms. They're implicitly shown by corners. Okay, and then we want 1, 2, 3, 4, 5, 6 hexol, which means that we want an alcohol group coming off every single one of those carbons. Now, this is more complex than you might think, because every carbon is not only going to have an alcohol group coming off it, but it's also going to have a hydrogen coming off it. Okay, now why is that complicated? Well, if you imagine that this ring of 6 carbons is sitting perfectly within the page, then off each one of these carbons, so let's just take as an example this carbon here, which I'm now circling in blue. Off every single one of these carbons, you're going to have an alcohol group coming off and a hydrogen coming off. Now, one of them will have to come off coming out towards us. Okay, so one of them will come off at an angle like this, coming out of the page towards us, because remember, these bonds we're assuming are in the plane of the piece of paper. The other one will go into the page away from us, okay? So the question then is, which one is going to do which? Okay, so really this is a question of optical isomerism. Every single one of these carbons is going to be a chiral center, basically. Well, it's difficult. It's more complex than just having enantiomers because of the ring structure. But there are many optical isomers of this molecule, basically, because, to give you a simple example of why there are multiple optical isomers, you could have all the alcohol groups coming towards us. Okay, so if every single carbon now have the alcohol groups coming out of the page towards us, then all of the hydrogens will be going into the page away from us. Okay, but then you could also have 3 alcohol groups coming out towards us. Let's say this one, and this one can have their alcohol groups coming out towards us, and then they'll have their hydrogen going into the page away from us. Okay, and let's say these 3 carbons have their alcohol groups going into the page away from us, and then the hydrogen's coming out of the page towards us. So now we've got 2 different optical isomers of inositol there. One where all of the alcohol groups are on the same side, and another where 3 alcohol groups are on one side and 3 are on the other. Okay, so you might now understand that there is quite a bit of complexity here in the optical isomers of inositol. Now, overall, there are 9 optical isomers of inositol if you work them all out. Okay, and you have to be good with visualization. You have to make sure that you're not counting the same optical isomer twice, basically. You have to make sure that you really can't superimpose one on the other. Okay, just by rotating it around. So, which optical isomer is present within cells, and thankfully there's only really one optical isomer of inositol that is used within cells, and the optical isomer of inositol that's used within cells is something known as myo-inositol, and I'm going to show you the optical isomerism of myo-inositol now. Okay, right, so we will start with this carbon here, okay, we'll let this be cut, well actually I'll do the labelling in a moment. This carbon is going to have its alcohol group coming out of the plane of the piece of paper. Okay, and the way that I'll show that is a bond that gets thicker towards the end. So this alcohol group is coming out towards us, which means that the hydrogen coming off this carbon will be going into the page away from us. Now I'm drawing a skeletal formula, so I don't need to show the hydrogen that's going into the page away from us, because remember, you don't show hydrogens coming off carbon atoms in skeletal formulae. Okay, then we're going to have the next alcohol group coming off this carbon here, and this will also be coming out of the page towards us. Okay, and then again, of course, the hydrogen will be going into the page away from us. The alcohol group that comes off this third carbon over here will again be coming out of the page towards us. Okay, so it's coming out towards us. Then, the alcohol group that comes off this fourth carbon here, this will finally be going into the page away from us, and I'll show this as a dashed line kind of going into the page. Okay, so this one's on the opposite side to these ones, and then of course the hydrogen coming off here will be coming out of the page towards us. This one round here, again, it's going to be coming out of the page towards us, like so. And then finally, the one that's coming off this final carbon, this will be going into the page away from us. So 3 are on one side of the ring, four are on one side of the ring, these 4 are coming out of the page towards us, and these 2 here are going into the page away from us. Okay, right. Now, this then is the structure of the myo-inositol optical isomer of inositol. Okay, now there is a specific way of naming up the carbons within the cyclohexane ring of a myo-inositol molecule. Okay, so you call this carbon here the first carbon, this one the second carbon, this one the third carbon, this one the fourth carbon, this one the fifth, and this one the sixth. So that's the way of naming up the carbons within the cyclohexane ring of a myo-inositol. So what you have to do is, if you've got a molecule of inositol, you have to try and superimpose it on this drawing that I've just given you. And if you can superimpose it, then it's a myo-inositol molecule, and then you need to label up the carbons like they're labelled up on this diagram. Okay, right. Now, why am I telling you all of this? It is going to become important for understanding why phosphatidylinositol 4,5-bisphosphate is called phosphatidylinositol 4,5-bisphosphate. What we're going to start off with then is we're going to attach this alcohol group that comes off the first carbon of our myo-inositol ring to the phosphate group that comes off our phosphatidic acid molecule. Okay, so let me show you this here. So here is our phosphatidic acid molecule, and now we're going to attach the inositol ring to the other side of the phosphate group here via a phosphoester link. And it is the alcohol group that comes off the first carbon of the inositol ring that is going to participate in this attachment here. So here is our inositol ring shown now in blue. Okay, and we have these 2 long-chain carboxylic acid molecules here, and our glycerol molecule is here. Okay, right. Now that's not complete yet. This is phosphatidylinositol that we've shown here, which is an example of a phosphoglycerolipid, but remember we wanted phosphatidylinositol 4,5-bisphosphate. Okay, so we now need to add phosphate groups coming off the 4th and the 5th carbon. Now these again are going to be attached on via phosphoester links. So what we're going to do is put a phosphate group there and a phosphate group there. Now if I had just drawn you this simple picture that we've got here, this cartoon, you would have then asked, why is this not phosphatidylinositol 3,4-bisphosphate? Why can I not label that carbon 3? Well, you now understand that there is a difference between the 5th carbon and the 3rd carbon of myo-inositol because of the optical isomerism. Okay, the 5th carbon here is wedged between two carbons which have their alcohol groups going away from us into the page, whereas the third carbon is between one that comes out of the page and then one that goes into the page. So there is a difference between the third and the fifth carbons of the inositol ring of myo-inositol anyway. Okay, so that's why it's not insignificant that this is phosphatidylinositol 4,5-bisphosphate, and it's why it's not called phosphatidylinositol 3,4-bisphosphate. This phosphate group here is off the fifth carbon, not the third carbon, and there is a difference between those two in myo-inositol anyway. So that then is the structure of phosphatidylinositol 4,5-bisphosphate. Now, it is a component of normal cell membranes. It's a pretty rare component of normal cell membranes. You have a little bit of it. It's basically got a large hydrophilic head here. So these three phosphate groups here, they interact very nicely with water molecules. So if I draw the phospholipid bilayer here, you'll have phosphatidylinositol 4,5-bisphosphate molecules sitting in the phospholipid bilayer like so. Okay. So you have the phosphate group that comes off the glycerol molecule along with the inositol ring and the other two phosphate groups that come off that, dangling into the cytoplasm basically, interacting nicely with water molecules, whilst we have the two long-chain carboxylic acids, which are extremely hydrophobic, shielded from the water in the hydrophobic core of the phospholipid bilayer. Okay, right, so that is phosphatidylinositol 4,5-bisphosphate, or PIP2. So now what we want to discuss is the reaction that all phosphoinositide phospholipase C enzymes, which is just another name for phospholipase C enzymes, are going to catalyze, which is that they are going to cut this phosphoester link between the alcohol group that comes off the third carbon of the glycerol molecule and the phosphate group that is attached to the first carbon of the inositol ring. So they're going to cut where I've shown this turquoise line here. Okay, and the two products then will be a glycerol molecule that has two long-chain carboxylic acids esterified to the first and the second carbon's alcohol groups. Okay, and the fancy name for that is a diacyl glycerol molecule. Okay, and the reason that that name makes sense is that we have a glycerol molecule as the backbone, and then we have two long-chain carboxylic acids esterified to it. Now, why then isn't it called dicarboxylic acid glycerol? Well, what does acyl mean? So let me just explain this in terms of a little diagram showing you esterification again. So here is my carboxylic acid molecule again. Here is my alcohol group of the glycerol molecule. When you esterify them together, you take off the alcohol group from the carboxylic acid group, you take off the hydrogen from the alcohol group, you bind these two things together to make water, and then you bind the carboxylic acid group's carbon to the oxygen of the alcohol group of the glycerol molecule, and this forms your ester link here. Okay, right. So what you will notice is that we don't actually add on the full carboxylic acid molecule, because we don't add on that alcohol group of the carboxylic acid group. We add on everything of the carboxylic acid group but the alcohol group that is an essential part of the carboxylic acid molecule. Okay, and this portion that is left over from the carboxylic acid molecule, which is everything but the alcohol group, this is known as an acyl group, basically. Okay, or the acyl group of the carboxylic acid. Now, it's not a molecule, it's a group, and the reason it's not a molecule is that it's not finished. It doesn't have a bond coming off this carbon here. It needs to be attached onto something else. Well, it hasn't got a final fourth bond coming off this carbon here. It needs to be attached to something else, such as the alcohol group, okay? And therefore, it's a group rather than a molecule. It's something you can attach onto the side of a molecule. Okay, so that's why these are called diacyl glycerols, because we have attached two acyl groups, which, remember, are these carboxylic acid molecules without their alcohol groups, onto a glycerol molecule. Okay, and for short, diacyl glycerol molecules are often abbreviated to DAG molecules. Okay, another name for diacyl glycerol molecules is diglyceride molecules. Okay, right. And the other product then that you're going to get is going to be the inositol ring with the free phosphate groups coming off. Now, the diacylglycerol molecule stays within the phospholipid bilayer when you create it, but this other product, once you've cut it off the diacylglycerol molecule, it's just going to drop into the cytoplasm. Okay, so let me show this here. So we've got an inositol ring in blue here, and specifically it will be a myo-inositol ring. Okay, and then you've got free phosphate groups coming off the first, the fourth, and the fifth carbons of that inositol ring, which I will colour in purple here. Okay, so this molecule is called inositol 1,4,5 trisphosphate. Now, if you were being really strict, you would call this myo-inositol 1,4,5 trisphosphate. Everyone usually drops the myo-inositol because it's the main form of inositol within cells. So if you're working in cells, it's just assumed that it is myo-inositol and you don't need to bother stating it. But if you were being really strict, if, for instance, you were talking to a chemist who might be working with other forms of inositol, you should say myo-inositol 1,4,5 trisphosphate. Okay, right. And for short, it's often abbreviated to I for inositol, P for phosphate, and then because we've got free phosphate groups, free there, IP3. And a slightly less short abbreviation is to use the I and S abbreviation for inositol, and then have a P3 like so. Okay, and then if you really wanted to be rigorous, you might say INS 1,4,5 P3 like so. Okay, right. Now, these two products then are going to have important downstream pathways. IP3 goes off and activates IP3 receptors, which are in the membrane of the endoplasmic reticulum. This leads to them opening, and they then release calcium from the ER, causing a calcium signal within the cytoplasm of the cell. Okay. Diacylglycerol, along with the calcium signal that's being produced by the IP3, activates protein kinase C enzymes. Okay, calcium also does a load of other stuff, but one of its functions, along with the diacylglycerol, is to activate protein kinase C enzymes, and then they go on to phosphorylate all sorts of protein targets. So lots of exciting signaling pathways involve this cleavage of phosphatidylinositol 4,5-bisphosphate, PIP2 here, into diacylglycerol and IP3. Okay, right. So we'll call it there for this video, and in the next video what we'll do is we'll begin the discussion of the phosphoninositide phospholipase C enzymes. We'll initially look at all the different 13 forms of phospholipase C enzymes, and then we'll focus specifically in on the phospholipase C beta family. We'll look at the different splice variants of each of the members of the phospholipase C beta family. We'll look at where they are present within their body, so the differential distribution within the body, and we'll then do a in-detailed study of the structure of these things and how they're going to be activated by G-alpha Q/11 family alpha subunits of heterotrimeric G proteins.